Energy is used throughout everything we do. To move, to digest, and even to think requires energy. Humans and other many living things break down energy in food and release adenosine triphosphate, also known as ATP, which is the main energy source in the body. Join me as we go through the process in which ATP is formed. Starting in the cytosol, let's start off with one molecule of glucose. Glucose is a sugar which is most commonly used as an example to examine the reactions and pathways involved in cellular respiration. Glucose provides the body with its primary source of energy. When sugar, starch, and carbohydrates are digested, they are broken down into glucose. Glucose is a 6-carbon sugar in a process called glycolysis, which occurs outside of the mitochondria in the cytosol. Phosphorylates being the highest energy phosphate bond found in living organisms and is involved in glycolysis and gluconeogenesis. Glucose is broken down into 2 molecules of a 3-carbon molecule called pyruvates. In order for this to occur, some energy needs to be invested. 2 ATPs are invested, and then 4 are released. This change from glucose to pyruvate is accompanied by a net gain of 2 ATP molecules and 2 NADH molecules. NADH is an electron carrier. Back when glucose is broken into pyruvate, 2 electrons were released and attached to NAD+, along with a hydrogen ion producing NADH. The citric acid cycle. 2 product pyruvate molecules are transferred to the inside of the inner mitochondrial membrane. In the mitochondria, the pyruvate molecules are oxidized. One of the carbons of each of the 3 carbon chain bonds with the oxygen gas molecule, which leaves the cell in the form of carbon dioxide. The remaining 2 carbon molecules are called acetyl-CoA. This process results in the formation of 2 NADH molecules, 2 FADH2 molecules, and 2 ATP molecules. When FAD is bonded with 2 hydrogen ions and 2 electrons, it becomes FADH2. FADH2 is an electron carrier just like NADH. The electron transport chain is where the NADH and FADH2 molecules end up. They end up in a chain of channeled proteins across the inner membrane of the mitochondria. These proteins send hydrogen ions from inside of the mitochondria across the inner membrane to outer compartments of the mitochondria. This creates a proton motive course. Once the protons are out, they want to get back into the inner side of the membrane. These protons are allowed back through a spinning protein called ATP synthase, which is found in the cristae. These proteins squeezed ADP molecules together with phosphorus ions, making somewhere around 34 to 36 ATP molecules. Now with all this ATP, the body can go above than doing its normal routine.